Climate change is already affecting our lives and will only become more impactful in the near future. All predictions show that decreasing carbon emissions will not be enough, and we need to actively remove greenhouse gases from the atmosphere. Current solutions, however, mainly view greenhouse gases as a waste product and treat it accordingly, opposed to as a potential resource. And that is where the DTU BioBuilders come into play. Our project, named PHEAST, aims to contribute to the transition towards a more sustainable and circular economy by utilizing the potent greenhouse gas methane as a feedstock for bio-based production. With respect to the greenhouse effect, methane is 25 times more potent than carbon dioxide and is being emitted into the atmosphere in large amounts, currently estimated at 600 million tons per year. Although it is a low-energy molecule, some microorganisms have evolved to live on methane as their carbon source. We want to take advantage of this and create a cell factory that can produce valuable compounds through methane valorization.
In methanotrophs, the first step in methane utilization is the oxidation of methane into methanol. This process is catalyzed by enzymes called methane monooxygenases, or MMOs. Most methanotrophs have a membrane-bound particulate MMO, or for short, pMMO, while others have a soluble cytoplasmic MMO, or SMMO for short, which is only expressed during copper starvation. When we had to decide which of these two enzymes to use, we did a comparison of their turnover rates. We concluded that the activity of pMMOs were much higher than those of SMMOs, and thus we decided to use pMMO. The best characterized pMMO comes from Methylotrophic capsulatus. It is comprised of 3 trimer units, A, B, and C, which are all dependent upon several cofactors such as copper and cytochromes. Since M. capsulatus is capable of producing this pMMO, we actually considered it as one of our cell factory candidates. However, our product range would be limited to proteins that do not need post-translational modifications, and during discussions with industrial stakeholders, we realized that we would be dependent upon co-cultivation to make a product of interest. For co-cultivation, one strain would be responsible for the methane to methanol conversion, while other strains would be responsible for making the product of interest. As pointed out during one of our interviews, co-culturing has a lot of drawbacks, especially the potential of out-competition between both strains. It was at this step that we decided not to do co-cultivation, but instead do all of our engineering in one unique strain that would be able to carry out all of the before-mentioned steps.
The choice of organism for our cell factory is a yeast strain Komagataella phaffii GS115, previously referred to as Pichia pastoris. K. phaffii is a commonly used workhorse in biotechnological industries as it is very efficient at producing recombinant proteins. Being a eukaryote, it is capable of making post-translational modifications of protein products, therefore will be able to produce more valuable and complex proteins compared to prokaryotic cell factories. K. phaffii is a methylotrophic yeast, meaning that it is able to grow on methanol as its sole carbon source. Transitioning from methylotrophic to methanotrophic requires implementation of a single oxidation step catalyzed by an MMO. Once methane is converted to methanol, K. phaffii has a great native alcohol dehydrogenase, AOX, that is strongly induced by methanol. Through K. phaffie's native metabolic pathways, the carbon flux is then directed towards biomass or energy production. The methanol-inducible promoter AOX1 is mostly used for heterologous protein expression in K. phaffii. Although it's a strong promoter, its activation requires certain concentrations of methanol, which can be sustained only using pMMOs, considering the fact that enzymatic activity of SMMOs is significantly lower. However, introducing a non-native membrane-bound protein to this yeast is a bit more tricky compared to SMMOs. To the best of our knowledge, recombinant pMMOs have not yet been successfully expressed in yeast. We saw this as an opportunity to develop new solutions for methane emissions, wherein we utilize methane as a novel resource for a yeast cell factory application. Our goal was then to engineer K. phaffii for methane utilization by recombinant expression of pMMO from M. capsulatus to build an efficient protein production platform for any product of interest.
The project plan consists of several different steps, which all require their own iteration through the design, build, test, learn cycle, also known as the engineering cycle. We decided to divide our project into 3 main cycles, each with a specific aim. For making our recombinant methane-utilizing phaffii strain, we opted for a genomic integration of the parts, as this allows for a more stable expression compared to plasmid-based expression. Additionally, it relieves the need for maintaining selection pressure during cultivation. For genomic integration into the phaffii GS115 strain, we opted for co-transformation of a CRISPR-Cas9 plasmid and a repair template in the form of customized DNA oligos. To increase our chances of successful genomic integration, we used a phaffii strain that is Ku70 deficient, meaning that it solely depends upon homology-directed repair to fix any double-stranded breaks in its DNA caused by our CRISPR-Cas9 complex. To use these plasmids, we had to identify potential integration sites and design associated guide RNAs targeting each of the 4 chromosomes in phaffii. To assemble our CRISPR-Cas9 plasmids with our site-specific guide RNAs, we used user cloning. In order to determine the cutting efficiency of each CRISPR-Cas9 guide RNA complex, we carried out an experiment with wild-type GS115 and GS115 Ku70 deficient strains, which were transformed with respective CRISPR-Cas9 guide RNA plasmids without providing a repair template, expecting that the Ku70 deficient strain would die from double-stranded breaks in its DNA. All of our tested guide RNA candidates had sufficient targeting frequency in the Ku70 deficient strains compared to the wild-type strain, allowing us to use them in the following steps. At this point, we were able to employ our designed CRISPR-Cas9 system to engineer phaffii.
Using genome-scale metabolic models, we assess strain viability and protein production abilities utilizing methane. Our model predicted that the modified strain would be able to metabolize methane and produce a product yield of the same level of our first considered strain, M. capsulatus. Our next step was to design repair templates for the 3 pMMO subunits A, B, and C. In their most elemental design, the repair template consists of an expression cassette composed of a promoter, a coding sequence, terminator, which are flanked by homology arms for correct genomic integration. Here we see how a repair template encoding one of the pMMO subunits was designed. The different parts of the repair template have to fit together in a predetermined manner. For simultaneous and directional assembly, we decided to go with Golden Gate assembly. We designed primers to add recognition site for type 2S restriction enzyme for all our parts. Our constructed parts were then run through a codon optimization software developed by GeneWiz. This was important for 2 reasons. Firstly, to eliminate any type 2S restriction enzyme recognition sites within the coding sequence, thus making it compatible with RFC 1000. Secondly, to increase the gene expression by increasing the translational efficiency of the gene of interest by accommodating the codon bias of host organism phaffii. The pMMOs at all points require high level of expression. GAP and TEF were the 2 constitutive promoters satisfying this condition. We chose TEF1 terminator as it's the commonly used terminator in yeasts. Furthermore, we added signal peptide to all the pMMO subunits to ensure the localization of the subunits to the plasma membrane as it is critical for its function. We chose a native phaffii signal peptide, originally guiding a glycosyl transferase to the plasma membrane as it yielded the best results from a cell localization prediction algorithm when fused with our construct. We tagged the pMMO-B subunit with a yellow fluorescent protein called Venus to verify the localization of the subunit in the plasma membrane. We chose to tag only this subunit as the domain of the subunit protrudes out of the yeast plasma membrane. Having integrated all the pMMO subunits in the phaffii strain, it is possible to monitor the fluorescence emitted from the Venus reporter on the pMMO-B subunit. This can be done either using a confocal microscopy or biolector with a Venus filter. We assembled the repair templates using Golden Gate assembly with PCR amplified fragments and transformed the yeast strain by electroporation. To test the functionality of the pMMO subunits, a knockout approach was used. We designed different guide RNAs to knock out 3 native genes, namely AOX1, AOX2, and ADH2, all involved in the conversion of methanol to formaldehyde. Knocking out all these 3 genes has shown to make phaffii unable to metabolize methanol. Thus, the methanol should get accumulated. The levels of methane and methanol can then be measured in the off gas during fermentation. Therefore, the effect of varying methanol concentration in a medium on the growth curve of phaffii were determined experimentally. We performed a bioreactor experiment with methanol concentration gradients in YP liquid cultures complemented by cell viability assay. Additionally, the GSM model was corrected and the theoretical highest possible biomass yield was obtained. To validate the predicted growth rate by our GSM model, the effects of the different carbon sources on the growth rate of the wild-type strains were tested.
The final main aim of our project was to establish pathways for recombinant protein production in our cell factory. To design our expression cassette for the soy-like hemoglobin production, we first developed an expression library using fluorescent reporters to monitor the transcriptional activity of different promoters. The choice of promoter was based on the efficient gene transcription and the ability to be induced by methanol. This approach is additionally well known for industrial usage. The promoter of our choice were the AOX1 promoter from the iGEM registry, 2 AOX1 promoter variants, 99 and 89% identical to the AOX1 promoter, as well as the FLD1 promoter, a native phaffii promoter for formaldehyde dehydrogenase. Similar to the previous cycles, a CRISPR-Cas9 plasmid and repair templates were designed for chromosomal integration of our genes of interest. Promoter efficiencies would be tested by fusing the promoters with a fluorescent reporter, enabling us to monitor the promoter activity as emitted fluorescence over time. Once the most active promoter would be identified, we planned to use it for expression of LEC hemoglobin to obtain the highest yield in our methanotrophic yeast cell factory. The expression profiles for various fluorescent reporters from the iGEM registry would provide the iGEM community with documented tools for expression of these in phaffii. For comparison matters, they were placed under the control of the same AOX promoter found in the registry. The expression profiles could be used to improve the dynamic regulatory model of protein production, thereby validating the predictions to experimental data.
As mentioned previously, one aim of the modeling was to estimate the potential yield of our system and therefore its economic viability. Another aim was to find possible pathways for the optimization of the protein production process. To obtain valuable results, we needed to model as many of the metabolic pathways of our phaffii strain as possible, so we decided to work with genome-scale metabolic models, or GSM for short. GSMs are based on genome-wide constraints. Considering we are in an n-dimensional space defined by the n reactions in our system, we start narrowing down the solution space by including constraints. Such constraints, for instance, could be the rates of certain reactions or the uptake of nutrients. Once we have included all known constraints, we start optimizing for growth and then protein yield. The values derived from this constraint space demonstrate to an approximate degree what we can expect from our working cells. Optimized for protein production at a particular growth rate, our model shows that about 1% of the total biomass produced will be our heterologous protein. Furthermore, based on literature reviews, we modeled the kinetics of the methanol-inducible promoter AOX1 as to determine the time dynamics of protein expression and the levels of methanol required to fully induce our system. We took rates from S. cerevisiae, as the literature about this yeast is just much more comprehensible. Moreover, we looked at studies investigating AOX activity as a function of methanol concentration and its main activating transcription factor MXR1. According to our investigation, we can expect our system to be fully induced at a methanol level of around 3% and expect full protein expression after 30 hours. And furthermore, we found out that glucose is the strong repressor of the AOX1 promoter, as are other carbon sources, and hence we will grow our yeast on methane alone after an initial growth phase on another carbon source. There was a cooperative back and forth between our theoretical modeling and the experimental results. After the initial model, we designed wet lab experiments to further and more accurately determine the time dynamics of protein expression in our system. Working with the GSM and the expression kinetics model, we spent a lot of time understanding how to set up these models and how to utilize them to their full potential. So we were wondering if we could create an interactive and intuitive app which would offer the possibility for future teams to model parts of their project with ease. We created a user-friendly app where it is possible to model the expression dynamics and yield of a protein of interest considering basic kinetics parameters. We also included some default values for these parameters for different organisms to facilitate an easy start for the user.
iGEM requires a lot of consideration for managing any risk to ourselves, our colleagues, community, or the environment. Even though our project does not involve working with any pathogenic organism, we still have to be aware of the toxic chemicals we are working with, as well as to prevent the spread of GMO or antibiotic resistance. The genetically modified phaffii GS115 Ku70 strain is incapable of doing non-homologous end joining, making it inferior to other organisms found in nature. The inability to repair DNA without repair template will mean they will most likely die if released into the nature. Disabled E. coli K12 derived strain like DH5 alpha can only survive under controlled laboratory conditions. The strains we are working with does not pose any threat to anyone in the lab as they are all non-pathogenic. For selection, we have used 3 resistance markers, namely chloramphenicol, ampicillin, and NTC. We are aware resistance markers, if let into the nature, can increase the threat of the antibiotic crisis. All measures have been taken and our final phaffii system does not have any antibiotic resistance as the CRISPR-Cas9 plasmid will be lost. The risks were considered throughout the project and revisited for any dynamic changes. For future validation of our methane-consuming cell factory, we have to be aware that methane is an explosive and special care has to be taken while testing it out as a substrate in a fermentation process.
Who could make use of this cell factory? We initially thought that in order to broaden the impact and effectively reduce atmospheric methane, we could run smaller scale fermentations on-site at sources of methane emissions such as agriculture. After successive interviews with political stakeholders, however, it became clear that using a transgenic strain like ours would not comply with EU GMO laws. Nevertheless, implementing it in an industrial setting in the biotech industry would not conflict with EU regulations. And therefore, going further, a solution could be engineered as to capture the methane at critical sources such as dairy farms and ferment it elsewhere. Moreover, these interviews and having a public discussion at a Danish political festival encouraged us to communicate projects like ours also as scientists in order to facilitate a more rational debate on GMOs.
Having had the opportunity to design the cell factory, our next steps would include testing the strain's newfound ability to assimilate methane and produce recombinant proteins of interest. For this, we would start by observing our strain's performance in pilot-scale fermentations before moving on to larger U-loop reactors. A U-loop reactor specializes in dissolving gaseous molecules in liquid cultures, which would help overcome the bottleneck that is methane's low solubility. For the future, there are many possible applications of such a cell factory, either as a production chassis where it would increase the sustainability of cell factory manufacturing, or by implementing it on-site of a biogas facility where it would aid in methane valorization. We hope that we could provide a good overview of the work we put into our project, and you can read about it in more detail on our wiki. This has been the DTU BioBuilders presenting our project, PHEAST.